The nitrogen cycle is said to be the most critical of the 9 planetary boundaries. Advances in agriculture have boosted production through the use of huge quantities of nitrogen, and this will eventually exceed the allowable limit. The use of nitrogen must be halved for it to be sustainable. Our project aims to reduce the amount of fertilizer used for crops and the amount of nitrous oxide emitted during cultivation. Nitrogen cycles through the environment, and most of this is carried out by microorganisms. We are engaging in research on nitrification and denitrification inhibitors for slowing down the conversion of nitrogen, allowing crops to absorb more nitrogen and suppress the greenhouse gas emissions. Soil consists of a mixture of minerals and organisms, and many microorganisms are found in it, although 99% of them cannot be cultured. So we rely on genomes and DNA to study them. But a lot of these microorganisms that can't be cultured probably harbor many treasures waiting to be discovered. Some of them undoubtedly have the ability to eliminate greenhouse gases, so we're striving to discover them. Specifically, soil is created over hundreds or thousands of years by minerals and microorganisms. But we're trying to do this artificially in a month or several weeks. We then inoculate the soil with the microorganisms we harvested to try and make artificial soil aggregate capable of reducing greenhouse gases. Until now, it was impossible for conventional soil science to achieve this. It will have a major impact that will change the foundations of this academic field and change everything we take for granted regarding the use of microorganisms. Rhizobia represent one of our achievements. There are nodules on the roots of the soybean plant inside which the rhizobia form symbiotic relationships with the plant cells. These are cells that have been infected with rhizobia. Here, nitrogen gas is converted into ammonia. These symbiotic relationships are unique to legumes. This is known as structural biology, and we make use of protein chemistry to try and develop nitrification and denitrification inhibitors, which will contribute to agriculture and protect the environment. The soybean, too, is modified to only form nodules with the rhizobia they've been infected with, enhancing the effect of infection. So we're studying soybean genes from all over the world to find beans with such traits. Moreover, methane is produced in rice paddies by methanogen. The Koshigikari rice variety produces large quantities of methane, but a rice variety was found that emits less methane. So we're currently trying to develop low methane rice. The mechanism behind this is currently being investigated. We'll move on to social application once this mechanism has been elucidated. I want to spread the use of microorganisms in nitrification and denitrification inhibitors based on evidence to mitigate greenhouse gas emissions. There are also bacteria capable of eliminating nitrous oxide or methane among the 99% of microorganisms that can't be cultured. We're asking elementary school students and the general public throughout Japan to help us find them. The initiative, known as Citizen Science, will study how much nitrous oxide is being emitted from soil and the microorganisms found in it. We've received a huge response, and that's become a driving force behind this research. I want to keep this virtuous circle going. Prevent global warming by relying on soil microorganisms to reduce greenhouse gas emissions. Cool Earth via microorganisms.